All right. So, welcome everyone. Welcome to today's workshop where we're going to talk about image segmentation and 3D analysis. My name is Ronel Sikat from the KAUST Visualization Core Lab. So, yeah, welcome to those who are here in person, and we're also live streaming the workshop today. So we have in-kingdom participants who are attending via YouTube live stream, which means the whole session will be recorded. So you can get back to the content later on. So if you miss a detail today, that's fine. You can go back to it later, review it, and follow at your own pace later on, right? So again, welcome everyone. Yeah, so before or during the registration, I put there a question about what you are interested in learning, what are you expecting to learn in today's workshop. And I highlighted here some of the keywords that we will touch on today. So of course, segmentation and analysis, it's there. But it's not only that, we'll also touch on a little bit on visual representations or generating visualizations for your data, which you can use for your papers. We're going to use Avizo. We're going to measure stuff. We're going to transform or convert your data from image to something else, let's say a 3D model, or your analysis converted into a CSV file that you can use for something else. We're going to work with biological images, so biological samples, but also non-biological. So we have rocks, and some people talked about membranes. So we're going to look at a membrane as well, in particular pores, right? So this is a very common theme here in KAUST. So people are interested in analyzing pores. So we will touch on that as well. And finally, deep learning. So we will look at some deep learning tools that can help you with image segmentation. All right, so throughout the workshop, I want you to keep this question in mind. And hopefully by the end of the workshop, you have some sort of answer. And that is, how can you use Avizo to visualize, process, segment, analyze, or transform your data? Right, so we'll focus on segment and analyze, but there's other things you can do with Avizo. It's super powerful. So I want you to keep that in mind. And later on, hopefully by the end of the workshop, you have some idea. If you don't know the details, that's fine. You can always approach us in the Viz Lab to help you. All right? So keep this in mind. So here's the overview of the whole kind of workflow that we will do multiple times in different ways. Because this workflow depends on the type of data you have. But basically, we start with your image stack, right? So that's the input data. And usually you get this from your microscopes, either using the facilities here in the imaging core lab. So you have your microscopes, we have CT scanners, could be a simulation, right? But bottom line, we start with either an image or an image stack. So it could be 2D or 3D image, as you can see here. And our first goal is to do segmentation. So what that means is, in our data, we potentially have one or more materials or objects that we're interested in analyzing. So the first step before we can do analysis is to label the data, right? So that means for each pixel or voxel in my data, I need to assign a label to it. So in this example, this is like an image stack of sand particles. So the bright white regions is sand, and the dark region is, let's say, air or void. So we use some segmentation steps to get to this label image, right? So here we have, let's say, the dark blue is sand and the light blue is air, right? So we do some steps from here to there. Depending on your data, it could be an easy process, could be complicated, right? But after we have the label image, the next step is to do analysis. So now that we have the labels, it's easier to run some algorithms that would measure things for us. For example, we can measure, like, within our data, or how much of the volume of the data is sand and how much is air, right? So this is just one simple example. You can do other measurements, like what's the distribution of, let's say, volume or size of the particles in my data. Could be pores as well, right? Yeah. So this is the, let's say, the workflow that we will work with today. And for that, we will use Avizo, right? So this is the software that we will focus on today. So in particular, version 2024.1. And Avizo is a commercial software, which means you have to pay for it. But luckily for us who are here in KAUST, we have a network license, which means as long as you're connected to the KAUST network, you should be able to access Avizo, right? And if you've checked out the web page for the workshop today, you should have found the link to the installer from the IT website. So just install it, follow instructions. I have some people here ask about the installation of the deep learning environment. So by default, or I'm not sure, it depends on the system, it gets installed in the P drive. If that happens to you, it's fine. Let's deal with that during the break, all right? All right. So before I get into all the Avizo stuff and segmentation, I just want to introduce first the lab where I come from. So that's the KAUST Visualization Core Lab, or KVL. And we are just one of the many labs in the Core Lab. And we provide state-of-the-art facilities and expertise in different fields. So we have chemistry, biology, supercomputing, and many others. And in particular, the KAUST Visualization Lab, or KVL, provides expertise in data visualization and data science. So we have a scientist who is focused on information visualization. We have some scientists, so Tom, my colleague, James, who's managing stuff in the background, and me, we're working on scientific visualization and different specializations. And we also have data scientists who help KAUST researchers with data science, machine learning, deep learning stuff. So aside from providing like basic support, like answering simple questions, basic consultation, we also collaborate with KAUST researchers and in-kingdom partners where we work on like bigger, more long-term projects. And in most cases, we are included as co-authors, let's say, in any publications that come out of the research. So that's a possibility. Aside from the expertise, we also provide facilities. So if you walk along the spine across the spine, just from here, actually, you would see like all the big displays. So we provide you access to that. So we have big 2D displays, we have large-scale virtual reality displays, but also head-mounted AR and VR devices. And once a year, we actually provide a workshop that discusses how the VisLab can help you with using augmented reality and virtual reality for data visualization. And yeah, if you're interested in taking a look at all the services we provide in the VisLab, you can check out our website. Or if you have a specific question, you can send us an email at this email address, okay? All right, so this is gonna be our schedule for today. So yeah, next I'll go to the introduction to Avizo, just because for most of you, I would imagine it's gonna be your first time using Avizo. So we're gonna do a quick overview of that, just to give you a feel of how to use it, how it works. And then we're gonna look at basic segmentation and analysis, and later more advanced examples, okay? All right, so let's move on to Avizo. Before I get into Avizo, I usually give this slide just to clarify some important concepts. So if you look at your image data, right, under the hood, it's just a bunch of numbers, right? So it's the bunch of numbers that came out from your imaging device, right? So it's what you see on the bottom left. So you have a 2D or 3D matrix of numbers, right? And this is your data. Now, in order to see something, see the patterns, we usually apply a color map, like you see on the bottom here. So this is a very typical color map. It's just a grayscale color map. And this means we assign a color and transparency for each number in your data, right? So for example, black or darker colors are assigned to lower numbers, and white assigned to higher numbers, okay? And by using that, by applying color maps, then it's easier to see the features in your data. Like in this example, now you see the edge clearly, right? Versus if you just look at the numbers, I mean, it's basically more difficult for our brain, right? So yeah, so you apply color map, and sometimes you want to try different color maps that could enhance other features that you want to show, but you're not changing the underlying data, right? So just keep that in mind. Now, when we do segmentation, we want to create a new image, basically, right? Where the numbers now correspond to the materials, right? So in this case, the number 1 corresponds to, let's say, air, and number 2 corresponds to sand. And if you have more materials, the numbers. I mean, of course, you can use different numbers, but just for example, right? And 0 is usually assigned to unlabeled pixels or voxels. Okay? So our goal is from your data to get that label image. And once you have the label image, now you can count, right? It's easier to count, and that's your analysis, right? Counting, measuring stuff, and so on. Okay? Any questions so far? Perfect. Because now we're going to move on to Avizo, all right? Let me just switch the camera. All right, so now we will go into Avizo. So I'm gonna be sitting here because my laptop's here. So when you open Avizo for the first time, by the way, for the first session until we take a break, I suggest you just watch what I do, and then later on you can follow with the hands-on stuff. My goal here is to give an overview, give you a feel of how to use it. Because if you start following right away, I mean, it can get confusing and difficult to get a good big picture. So for now, just watch and try to understand the, let's say, the framework, how Avizo works, all right? So when you open Avizo, you get a window like this. So you have these red icons. I would say, like, in the beginning, these are the most important parts. So you can have open data, open project, or just start with a blank project. Let's just open a data set. So I will click open data. And if you open, or I will open the datasets folder that I shared with you. So these are the datasets inside. I will open the sand dataset. So if I open that, okay, it's too dark there. But if you look at the TV screens on the left and right, you will see that, I mean, let me open one image. Oops. Yeah, you will see that there's a bunch of images with the slice number in there. So it so happens that my data is given as a bunch of 2D images, right? So I just do Control A. I want to open all of them. And in the beginning, Avizo will ask you for some basic information about your data. So you can give it a name, the units you want to use for measurement, the voxel size, right? So this information comes from your imaging modality, right? So for now, I'll keep everything default. Click OK. All right. And now comes the interesting stuff, right? So there's, let's say, 3 very important windows. So the one here on the upper left is the project window. And here you're going to see a bunch of icons, right? So each icon here corresponds to data objects, right? Like this one. So this is my data. Data objects and, let's say, visualization and compute objects, okay? Like this one here. So let me just delete this one first. Okay. So right now, after I open the data, Avizo created this object. And if I click on this object, on the bottom left, you're going to see some of its properties, right? So what's the size, the data format, memory size, and so on, right? So that's what the property window does. Whatever I click in here, it shows me the property of that object. So now again, for my data, I want to take a look at my data, right? So now I'm going to click this white, I'm not sure, yeah, it's clear, this white circle button on the right of the object. And it will show me a list of other objects that I can connect, right? Which can be compute or visualization. So for now, I will attach an orthoslice. Okay. And this orthoslice, because it's a visualization object, it actually creates a visual representation of my data on the 3D view over here on the right. Okay. And you're going to see, I mean, I can move it. It's just a 2D slice of my data, right? And again, if you look at the property window, if I click on the orthoslice object, I'm going to see the properties here. So you can change the orientation, how you slice the data, and the slice number, right? So you can basically get a 2D preview of your data by inspecting this. Yeah, it's a bit too dark on the big screen. But yeah, basically, for example, you see the color map over here. So you can change that as well. So I won't go into the details, but basically you can adjust the range here to change the contrast. You can change the color map. If you click here, you can use a different color map, okay? Depending on what you want to see. For now, we'll stick with the grayscale. So yeah. So this allows you to see your data in 2D. So something helpful in the beginning is also, so if you click on your object, on the top here, you're going to get some suggestions, right? Like some frequently used modules. So I would just add a bounding box. So from the name itself, it draws a box around the boundary of your data. So it kind of guides your visualization. Like it tells me from the top and bottom parts of my data. Okay. So yeah, that's the super basic like visualization part of Avizo in 2D. And now let's say I want to see my data in 3D, right? So one of the most common modules to use is the volume rendering, right? So if I now click volume rendering, it would give me a preview of my data in 3D, okay? This can be tricky, right? So Avizo by default assigns the color map for you, or in 3D it's called the transfer function. So depending on how good Avizo kind of figures out what's in your data, you're going to see different things here, right? So let me just quickly demonstrate that. So this is the transfer function. Again, it's a bit out of scope of this workshop, how to work with this, but you can approach us later. But if I change this transfer function, you're going to see different things, right? So this is assigning different, let's say, color and transparency, opacity to the data, right? So for example, here, it's hiding everything except for the high density stuff, okay? But yeah, you can play around with this later on, okay? But this now gives me a really nice preview of my data, how it looks like in 3D, right? Without doing any like special processing, okay? And just like the 2D, the orthoslice, you can also change the transfer function, right? Depending on what you want to highlight, right? So for example, this one kind of highlights the high density region, right? So depending on the image you want for your paper or for your analysis, you can design the transfer function, okay? All right. So that's kind of a little bit like basics of Avizo and visualization, right? So we already did some sort of visualization. If you just want to show in your paper how the data looks like in 3D, you're done, right? Actually, I can show you quickly. how to take a snapshot, right? So there's an icon here, like a camera icon. If you just click that, yeah, you can save the 3D view as an image, and you can use that in your paper, right? Later on, if there's time, I'm going to talk about how to create a video for, yeah, that you can also sometimes use to present your data. Okay. All right. So that's it for like basic visualization stuff and basics of Avizo. I hope somehow you have a feel of Avizo. So you create these objects here. So you have your data objects and visualization objects. So there's also these green or blue buttons here that you can allows you to turn them on and off in the viewer. That's pretty helpful. All right. So now we'll get into segmentation, right? Which is the like one of the main goals we want to do today. So for the first example, let me just go back to interesting, the shading changed, but anyway. So let's say my research involves measuring the 3D volume of the sand particles in this scan, okay? So how do I do that? So first I have to segment the sand particles, right? To label those pixels or voxels, and then do the measurement, okay? And right now I will just go through the steps, okay? Okay, so I would actually go here. And one of the first tools that I would use, and I recommend you also do, is the interactive thresholding, right? So again, for my data, I click this circle button, and from the list here, I see interactive thresholding, or I can type it here as well. All right, so when you create an interactive thresholding module, there's a 2D slice that automatically shows up. It's basically an ortho slice, but so you can change the slice number as well. But there's this blue highlighted region, right? And what's that? So that's actually a preview of the thresholding result if you use the parameters here. I'm not sure. Yeah, so you can see it here. So what's thresholding, right? So thresholding is saying I have a minimum and maximum intensity value that I'm interested in, right? So everything that's between these values, I want to assign a label of 1. Everything else is 0, okay? So the blue ones that I highlighted here will be assigned to 1 if I click apply, and everything is 0. And you can actually adjust that parameter here, right? So of course, if I set this to 0, it will select everything, right? Because everything is between 0 and the maximum. But what I want is just the sand particles, right? And just from looking at the data, I mean, the intuition is that the brighter colors correspond to sand, right? So the higher intensity values correspond to sand. So if I just figure out like the correct threshold value here, I should be able to get separate the sand from the air, right? So like this, for example, right? If I click apply, a new data object gets created here. And that's the label, actually. So you already did segmentation, right? So this is a preliminary segmentation. So if I turn off now the interactive thresholding, click on the result. Again, click here on the circle. Or in this case, I see here the module I want, which is voxelized rendering. Yeah, let's say this is just a more optimal way to look at label data, voxelized rendering. And now you see my segmentation result, right? So this is it. We already did segmentation, right? So this is, let's say, the simplest way to segment something. And now this is my sand data. But as you can see, it's quite noisy, right? So there's a lot of like unwanted particles over here. And you probably saw that in the interactive thresholding, right? You see these, like these outlier, like blue particles. And maybe that's actually sand particles, then it's fine. But in this case, I know that it's noise, right? It's noise from the, let's say, imaging modality. So what I want to do is apply a filter to my data. So I will pre-process the image to reduce the noise, and hopefully that will give me a better segmentation result. So let's do that right now. So I'll turn off the thresholding. I go back to my original data, and I will open a filter sandbox. Okay, so the filter sandbox will show me again a slice of my image. So the filter sandbox allows me to try different filters or different, let's say, image processing algorithms to apply to my image to, let's say, reduce noise, right? So I can specify a region where I want to see the preview. So if I click here, I can choose different filters. In the interest of time, I will, because I know the median filter would give me a good result, I just pick that. So you see here, it's not clear on the big screen, but if you look on the side screens, the median filter kind of reduces the noise. And yeah, so the idea is to play around with the parameters here until you find a good filter to use, and then when you're happy, you click apply. So that creates a new version of your data that's been filtered. And now I can move the input to the interactive thresholding, to the filtered one. And I can go back to interactive thresholding. And now if you look here, I hope it's obvious that even with the same threshold value, you don't get those tiny like outlier particles anymore, right? Because we filtered them out. That's noise. So now if I click apply again for the interactive thresholding and look at the result, so now you see it's much, much cleaner, right? So you can clearly see the sand particles without the noise. Okay, so I hope that's clear, and I hope the mental model is there. We have the original data here. It's still there, by the way. It's still noisy. Then we use the filter sandbox to find a good filter to use to reduce noise. We found one, we applied it, then we get a new version of our data. We reapplied the interactive thresholding, and finally we get a good segmentation over here. All right. Now that we have the segmentation here, now I want to count, right? So as I said, I mean, for my research, I want to, let's say, count how many particles are there or measure the volume, right? The 3D volume of the particles. But right now, each particle is kind of stuck together into one piece. So I want to separate them, right? So in Avizo, I mean, there's a module called separate objects. First of all, I know because I've used this before, I don't expect you to know all the modules right away, right? I mean, it takes experience. There's also like a very nice user guide. If you go to help, if you click user guide, it brings you to this website. You can search for keywords, right? Like something you want to do, and it will tell you which module is helpful for you. Okay. Okay. So now I'm gonna separate objects. Click apply. And if I now look here, it's not super obvious, but it creates gaps between different objects. And the next one, which we will use a lot today, is the label analysis. So let's just try that. So the label analysis generates two things. It generates a table with different measurements of each of the object here. And it also creates a new label where now each object is given its own unique ID, right? So it's not perfect, but I hope you get the idea, right? So now each particle has been separated, and it gives me the measurement for each, right? So For instance, you can again take a screenshot of this for your paper. And now you have this table with the 3D volume, surface area, and the central coordinate. And if you want to put this table in your paper, you can click this button here to export. So you can export as CSV, for example, comma-separated variables or values that you can open in your text editor or some spreadsheet application. Okay? So, yeah, we have to take the break now, but hopefully this kind of initial session gave you a feeling of how Avizo works, right? So you create these objects, you set the input, output, you tweak the parameters for the algorithms, and you build your workflow, right? So we started from our original image, this one, and we ended up with our segmented data and the analysis results, okay? So this is a super basic example. And again, just an overview of Avizo. After the break, we're gonna look at more examples, okay? So let us take a 10-minute break. All right. We're back from the break. So before I proceed, I just want to make a clarification for those who are here in person trying to install the deep learning environment for Avizo. If it gets installed in a folder where you don't want it to be, like the P drive, you have to set the environment variable home, H-O-M-E, to the folder where you want your deep learning environment to be installed, and then close Avizo, reopen, and then try installing, okay? So let's see later. We still have one more session and then another break before we get into the deep learning stuff. So let's have another look after this. All right, so we've had a look at the basics of Avizo and like a super simple segmentation. So now I want to give you like, let's say 5 minutes, open Avizo, and I prepared in the data folder 3 super simple data sets. So actually it goes from easy, intermediate to, let's say, a bit more advanced, where you can do like exactly the same thing that I did. I mean, you don't have to do it perfectly. I just want you to get the feel of, you know, opening a data set, creating modules, and so on. So look for the spheres, sand, and foam data set in the data folder for those who are here and has Avizo. So I will actually, so you can pick, it's up to you. I suggest those who have experience with Avizo open the foam. For those who are starting, the spheres is super easy. Basically, you just apply a thresholding. And yeah, feel free to do it now. And for those who just want to watch and for the live stream, I will do the, let's say, spheres, okay? Okay, so now we're back to Avizo. I can close this and open, or I can just go to new project and discard everything. So I'm left with an empty project again. I will open data. So again, to my data folder, I will open spheres. Yeah, just set the units. Again, after opening the data, you get your data object. And now I'll just go ahead and do volume rendering. And as you can see here, it's just a bunch of spheres, right? Which is a super easy data set. And if I look at the ortho slice, yeah, it's literally just a bunch of spheres, which is good. So imagine these are your pores, or again, sand particles, or yeah, pores in your membrane, pores in the rock, etc. And in this case, so I have this grayscale color map again. So the white regions are like high intensity value stuff, high intensity value materials. So again, from your object, this one, it's clear to me that I don't have to apply filtering. So I'll just go ahead and do interactive thresholding. And Avizo is pretty smart. So usually it suggests like threshold values for you to use. So here right away, it gives me a good threshold to use. And if I just click apply, it creates the correct label. So again, voxelized rendering. And there you go, right? Super easy. Again, it depends on your data set, right? Like this is a synthetic data set, so it's super easy. So I have the labels. Again, I want to do analysis on this. So you can do label analysis. And here, since the objects are already separated from each other, I don't have to run separate objects module. So I just click label analysis in 3D and click apply. And just like before, we now have the individually labeled particles and the table that shows me the analysis, right? So you have the 3D volumes and the area. Just some cool like things that you can do here in the table. So you can click a column and let's say sort them ascending or descending order. You can check out the histogram, right? So to get a feel of the distribution. You can use this for your paper as well, right? Like if you're trying to report on the distribution of the sizes of the particles or the pores in your data, you can use this. So yeah, that's again another super simple example for segmentation using thresholding and basic analysis. Okay. All right. So did anyone try the foam data set? Not really. How about the sphere? Nice. How about the sand? Did you try it again? So the sand one is and the foam are good because in order to get a good result, you'll have to use at least some filter to denoise. Okay. All right. So any questions with the basic stuff? Good. No questions so far. So I'm gonna move on to the next. Okay. What is it? It should be there. So are they in Kaust? Okay. How do they have Avizo? No. So, okay, there's a question that they don't have the separate objects module. There could be several reasons. So one, if you're outside Kaust, first of all, you shouldn't have access to Avizo unless you're paying for it. If you're inside Kaust, we actually have a kind of a limit, let's say. We have, the way the license works is we have a number of tokens that gets distributed or that gets used whenever a user opens an Avizo instance, and depending on the modules you use as well, right? So if there's like, I don't know, 50 users in Kaust who have an Avizo window open, then maybe no one can open it anymore, right? So there's a certain limit. So it's possible that we've reached the limit somehow, but I don't think so. But it's not about the version. No, this is a very old module, I would say. Yeah. So, yeah, but if they still want, I mean, they can reach out, send an email, and then we can take a look. But if they're outside Kaust, yeah, we can, yeah, they have to check with their, anyway, so now we're gonna take a look at CT scan of a coral. So a few of you wrote that they're interested in doing measurements, measuring the surface area, and converting their coral CT scan to 3D models. So we're gonna do that. So this data set is from Eleonora Re and Domingo Sanchez, who's the CT scan guy. So let's go ahead, back to Avizo, new project. Yeah, so let's just open data. and open the Coral dataset. So one thing you'll notice here is that the data is just one file, right? Instead of multiple 2D files. And it's actually already in an Avizo format. It's .am. And the CT scanner here that's in the petroleum engineering lab can actually save the CT scan results into an Avizo format, right? So you don't have to do any conversions in between. And that's what I have here. So I'll just open that Coral dataset. Again, you can use an orthoslice to take a look, but it's for 3D data. It's nice to use the volume renderer. And here again, Avizo gives us a really nice initial transfer function, and it shows us the coral body, right? I believe this was a dead coral, and yeah, you can see basically the general shape of the coral. Okay, and what we want to do is extract segment the coral body and measure the surface area. For instance, if you want to measure, let's say, coral growth, this is kind of a process that you can try to do, right? So you can scan a coral before, then let it grow for a few years, I don't know, and then rescan it again. Probably not by CT, maybe some other method. But once you have both scans, you can generate the 3D model from both, or measure the surface area from both and measure how much it grew, right, in terms of surface area. So for today, we'll just do it for one coral sample. Okay, so let's do that right now. So just looking at the slice, right? Again, just from experience, and I guess by now you can tell that just by using thresholding, you can probably get a decent segmentation from this, right? I mean, it's actually super easy to get a preview. So if you just click the coral data again, interactive thresholding, again, it gives you a suggestion, right, for the threshold value. I'm not sure if you can see it here, but there's also kind of a histogram of the data here that can guide your setting of the threshold value. So for example, I can move the threshold value going here. Try to get, because I'm just interested in the surface area from the outside of the body, right? Not the inside. So I want to get the insides as well. So I can adjust the threshold value, let's say something like this. So you can preview the slice. So there's still some holes there, but we can fix that in a next step. You can also, if you want, apply a different filter. I'll skip that for now. But you can apply a filter that could maybe smoothen out your data or reduce the noise. For now, let's say I'm happy with this. Okay, so now if I look at the segmentation, it looks like this, right? Which is super good, considering we spent I don't know, less than 1 minute on it. But if you're gonna measure surface area on this one, on the outside surface area, you're gonna get a slightly wrong result because if you look at the inside, okay, if I attach an orthoslice, you're gonna see there's a bunch of holes, right, inside the label, right? So you're gonna see these things. So here. So if you're not concerned about that, fine, you're done. Just apply a label analysis, and then you get some surface area. But let's say, for instance, you want to fill up those holes. If you don't know what module to use, search, right? In this case, I kind of know what keywords to use. I would say if you search for something and then you don't find anything interesting, try different keywords, right? And then if you can't find anything, ask us first. Then we might know what module for you to use. So in this case, it says, okay, closing. Of course, I mean, you'll have to read the description, etc. But for now, let's say I know I'm going to use closing. And yeah, so the idea of closing, just quickly, is there's a certain shape. Let's say in this case a disk, and let's say I want to do it in 2D, that has a size of 3 pixels, I think, radius or diameter, that it will slide around the images, right? And if it sees that the regions around the disk is full, it will fill up everything that's inside, okay? That fills up the holes. So if I apply that now, you're gonna see the result looks like this, right? And you can see there's still some bigger holes left. So the smaller holes are filled. So if I want to fill those bigger holes, I would adjust this parameter here, size of that disk, and now I fill it up, right? So it's up to you. You're the expert. You should know at some point like what parameters work best for you, depending on your needs. And now, if I look at my segmentation in 3D, it looks kind of similar, but it looks more beautiful, to be honest. But if you look inside, you should see that most of all of the holes should be filled. Yeah, beautiful. And now, if you do a label analysis on this, you should get the correct surface area. Yeah. So here, you get 2, because there's probably like one small island that's left there. You can ignore that, but yeah, I think this is the bigger one. So this is now the surface area of your coral. Okay, so now let's say you did that and you want to export your segmentation result into a 3D model, right? So you want to convert this into a 3D model that you can use for another application. So in some cases, not for coral, but for membranes, I had a case where they wanted to export the data into a 3D model so they can plug it into a simulation software, right? So all I have to do is click on the segmentation data, click on this circle icon here, and create a generate surface module. Yeah. So for now, we'll keep the default parameters. If I click apply, that's it. Now we have a surface here. That's a 3D model. And if you click that and attach a surface view, now you should see your 3D model here. So what's the difference? Now, this actually consists of just a bunch of triangles, right? So if I just yeah, so I can change the shading. So if I zoom in enough, you would actually see the triangles, right, that describe your surface, okay? And depending on your simulation software, you might have to apply post-processing to this mesh. So some software need like very regular triangles and maybe a certain number of triangles. So you can click on the surface here, and Avizo has some modules. Yeah, so triangle quality, triangle distortion, etc. Yeah, you can look up the documentation, or you can approach us if you need help with that. Okay, but for now, just one more step. Of course, now you have the surface. You want to export it. So again, just click on this circle part here, and then click this button, Export Data As. And if you click the drop down, you can see the different formats that you can export to, right? So you have OBJ, for example, COMSOL, STL. So there's different things. I would say just read the documentation on what format your other software needs. Okay. All right. So for now, yeah, that's it for the coral. I think Let me just check. Okay, we have 15 to 20 minutes for the next example. Any questions about this one? Yeah? Mm-hmm. Yeah, so sorry, I think I went a bit too fast. But just to quickly recap what happened. So we applied the interactive thresholding. Let me just turn this off. We applied the interactive thresholding to get kind of the rough segmentation, right? This. But we saw that it has holes inside. Okay, and we don't want those holes, right? Because if we're measuring the surface area, those holes will give us a bit of error. So I use this closing module to close the holes, right? So after closing, I get this version where if you look inside, the holes are gone, right? And from here, I apply label analysis. And the label analysis did the measurement of the surface area for me. Yeah, I have this number now. That's the area, surface area, and the volume of the coral. And this is what I can use for my paper, for example, right? Like, okay, this sample has this surface area. You redo this for the, like, after 1 year, whatever. You measure the surface area, and then you say, oh, the coral grew by whatever meter squared. And if you want to export the model or export the segmentation as a 3D model, right? For example, for simulation or other things, you use this generate surface module. So that generates this surface object. And if you look here, right, it's just a bunch of triangles now. And you can export this. If you go here, you can export this into different formats. And you can plug this into your simulation software or other things, okay? Yeah. Okay, so that's it for the coral sample. I'm gonna move on to the next sample, which is a rock. Yeah, pretty popular here in KAUST. So it's a basalt core. So it's a core sample. Again, from the CT scan facility here in the petroleum engineering department. So I will just open up that data. It's called basalt. Okay. So for this one, I'll use an ortho slice in the beginning. And just go through the slices. So if we look at the 3D, just to give you a better impression, it's basically a piece of rock. It's a core, because I guess they take the samples as a core, right? So it's cylindrical in shape. So it looks like this. Yeah, so it's a rock, right? So there's different things inside. There's pores. So if you just look at the slice here. So these dark regions here are the pores, which are just empty regions inside the rock. There's other things here. I don't know what they are. The domain expert would know. But one thing that the domain expert told me is that they're interested first about the pores, because let's say that tells you how much gas you can trap inside the rock. And then there's the like bright white region, which is the rock mainly. And then there's this kind of weird boundary thing that's in between the pores and the rock, which is apparently minerals kind of growing, or let's say growing from the rock towards the pores, okay? And what happens is, because of this mineral, the amount of gas, the amount of void you have gets reduced, right? And for them, they want to measure that, okay? They want to measure how much the mineral, basically the volume of the mineral, right? Because the volume of this mineral is the amount of gas that they're not able to store in the rock anymore because of the mineral. Does that make sense? So I would say in terms of the segmentation perspective, my goal is to segment two things. First is the voids, because that tells me how much gas I can trap in the rock. And the second thing is the minerals, because that tells me the reduction in the amount of gas I can store. Okay? So yeah, let's try that. I would start with the pores because it's easy, as you can imagine. And don't expect, I mean, when you open your data and try to segment your data, that's always this easy. We're just lucky, or maybe I intended it to be easy for this workshop, but it's not always easy. Okay. So anyway, let's start with this one. And again, I would start with interactive thresholding. And in this case, Avizo suggests this, right? And from what you can tell, it tries to segment the rock and the mineral together. Yeah. But that's when I don't want that. So I will change this. So I will change the range, say 0 to some lower value. So again, the preview gives you an idea what's getting segmented. There's other ways to do this, by the way, right? So, but let's stick with this for now. So now I'm getting this, and it's including the outside, right? Which I don't want. But I will show you a module that will remove the border, okay? So for now, let's do this. I click apply. And if I look at now, let me just turn this off. If I look at the result, I get this, right? So it's the outside plus the voids, okay? Let's say in this particular case, I'm only interested in the voids that are completely inside the rock. So I will remove the outside stuff. And again, I would want to remove the border. It just so happens that I know there's a module called border kill that if I apply it, I mean, you can do 3D or 2D. So what that does is, let me just apply it now. It checks the border, right? So the boundaries of your images, and it kills or removes everything that's attached to the border, okay? So if you now look at the result here, magically, it just gives me everything that's not attached to the border, okay? Again, this is not the only way to do this. There's another way. Maybe I can show it now. Okay, so let me just disable this. There's different ways. So another way to do it is to crop your result, right? So again, let's look at this. So from here, you can crop your result. And in Avizo, you can use what's called a volume edit module. Volume edit. And basically what you can do, you can specify a shape. In this case, cylinder, because I know I want to crop out a cylindrical region. And z-axis, yeah. So it kind of, gives you this shape. So you'll have to modify it, right? So you'll have to adjust the size, the position, that would allow you to crop the data, right? It's more manual, but it's fine. Like this gives you more precision depending on your needs. And then let's say I want to cut outside, I think. Yeah, and it gives you this result, right? Yeah, so of course, like I missed this part. So just from experience, and I mean, depending on your need, I would prefer the border kill example, okay? So this just shows you that there's different pathways to get to your result, okay? So for now, let's say I'm happy with this result. And I can already measure this, right? Yeah, I can already measure this in the sense of if I just want to measure how much of my whole sample is air versus everything else, I'm not sure if this will work. Let's try it. There's a module called volume fraction. If you apply, yeah. So it gives you the fraction of this, let's say, part of the data compared to the whole data set. In this case, it's, let's say, 1.8%. But, okay, this was measured with respect to the whole data, including the outside stuff, okay? So in order to get the correct fraction for just the cylinder or the core, yeah, maybe this is where the volume edit would make sense. I would, sorry. Yeah, this is more tricky. Yeah. No. So you'll have to, this is what I wanted to say. So the question was, if I applied the border kill anyway and use the volume fraction, won't it give me the measurement without the border? Not really, but there's a way to do it. You'll have to define a mask. So that's another like label field that just tells you what you're interested in. So in this case, let me, it's a bit more tricky, but let me do that later, okay? Because yeah, let me do that later. Just put a pause on that, because what I'm gonna do is I'm gonna segment the mineral, okay, and then the rock. Then we're gonna count. We're gonna do the volume fraction for all those three. All right. Okay. So we only have 5 minutes to do that. So I'll try to go through it fairly quickly. And I only can do this because I've tried this earlier. So now, okay, I want to segment the minerals. Right. So let me create a thresholding again. So here is now an example where just using the thresholding will not give me good results. Right. So, okay, I'm going to try to find a range of values that would give me just the mineral. Yeah. So it's actually not easy, okay? So if I do this, you know, I'm capturing the minerals, but I'm also capturing some of the rock. And this is where the filtering really helps a lot, right? So now I would go to the filter sandbox again. So let me just turn off everything. Yeah, so filter sandbox, you have to like put the box where you want to show the preview. So I can try a bunch of filters, right? And it will modify the image, right? My goal here is to apply a filter such that if I apply a threshold to the data, it's easier to get the minerals out. And there's a nice feature in the filter sandbox, actually, where if you click this preview type, interactive thresholding, and set the threshold range. Oops, sorry, I changed the wrong thing. Where is that? Ah, here, sorry, it's right here. Yeah, if you change the threshold thing, yeah, it actually gives you a preview of what the thresholding result would be if you've applied the filter first, right? So this is really good for just trying different filters and see how it improves or like worsens your result, right? So I can try different things. And of course, you'll have to play around with the threshold value here. So just by trying different things, trial and error, and a little bit of experience, I found that if I do this disk, I get something decent, right? I hope you see it. That now the mineral is kind of easier to threshold, right? It's not going to be perfect, and I mean, we don't have time to get like a super perfect result, but let's say I'm happy with this. I click apply. Yeah, we're running out of time as well. I click apply, and I apply thresholding here, and I can just copy paste the threshold that I had here. Can be a bit confusing, but again, you can go back to the YouTube video later on to trace back the steps. And now you can see, right, of course we have a different image, it looks different, but that's because I was focusing on the mineral part. And now if I click apply, I should have the mineral segmentation. Okay, yeah. So now this is just looking at the minerals, okay? And you can kind of get the impression that the minerals are surrounding the pores, right? So, which is good. And it's not perfect for now. We'll stick with this. And now what I'm gonna do, I'll do it quickly, is just segment the rock part. Maybe it's too confusing now, but yeah, we have actually 0 minutes. Let's try to do that super quick. And now I have just the rock. Ooh, this has the rock and minerals. Yeah. Actually, this has the rock and the minerals. Yeah. So I have 3 segmentation masks right now. Okay, so I have this, which is just the rock. I have this, which is the minerals. And I have this, which is the pores. Yeah, unfortunately, we don't have time. Should I do it? Actually, let me do it just for the minerals and pores. So there's a module in Avizo where you can combine results, right, from different processes. So for example, just to keep it simple, I'm gonna combine the mineral with just the pores. Okay, let's do that now. So I'm gonna create what's called an arithmetic module. This is super important. Arithmetic module. So this allows you to apply arithmetic or logical operations between labels. So I'm gonna set input A to the minerals and input B to the pores. And naively, I'm just gonna do A plus B times 2. Times 2. What does this do? Is that even correct? Let's take a look. I said naively because there will be overlaps. But if we now look at this result, yeah. So the pores are set to a value of 1, and that's the inside here that you see. And the minerals are assigned a value of 2. That's why I put a times 2. That's the minerals you see here. And you're gonna see some red parts, right? And that's because in some places there's gonna be overlaps, right? So we can fix this later, but we don't have time to do that now. But there's gonna be those overlaps, right? So maybe in your analysis, you want that, maybe no. But now, if I do a volume fraction on this one, it should give me at least the, it's a different table. Yeah. So now it shows me the volume fraction for, let's see. I think 1 is the pores, right? So it's 3.2% of the whole data, and the minerals is 1.7% of the whole data, right? As I said, this is not completely correct, but yeah, that's a first step. So we don't have time to show the whole thing, but hopefully you get an idea. And if you want to really do this for your project, you can always come to us and we can show you the whole workflow. Yeah, and this is something you can report on your paper, for example. Okay, so actually, before we take a break, I'll just do something super quick, just to demonstrate. And it's a pretty common thing you want to do. For your paper, for example, you want to show like a nice image of the rock with the pores, right? So you want to highlight the pores, let's say. So you can do like voxelized rendering for the pores and a volume rendering for the rock. And then you can adjust, let's say, the transparency of the volume rendering. So you can have something that looks like this, right? So it gives you, the volume rendering gives you a context of the rock, like the size and shape of the rock. And at the same time, it shows you where the pores are, right? And you can take a screenshot of this and put it in your paper. Of course, you can make it nicer, but this is one other cool thing with Avizo. You can kind of compose and combine different visualizations depending on what you want to show in your paper. Okay? All right, let's take now a 5-minute break. All right. So yeah, hopefully by now you have a feeling of what's possible in Avizo in terms of segmentation and analysis. I know I went a bit too fast maybe, but that should be fine. Again, you'll have the YouTube recording later on, and I will send the email with the YouTube link to everyone today, so you can go back and follow it. And yeah, now let's move on to, let's say, a bit more advanced segmentation and analysis. So I mean, before I go back to Avizo, as I said, right, the examples that I showed so far are super easy data sets. So the next data sets we'll look at, something like this. Yeah, so this is a cryo-EM data set, right, of a cell. This is more tricky, right? So even for a human, I would say this is pretty difficult to segment, right? Like if I ask you to separate the fibers or I'm not a biologist, but basically like the tiny spheres, it's not easy, right? And this is where more advanced tools would come in, and Avizo provides those for us, right? So there's different ways that machine learning and deep learning are used in Avizo. But before we can use those, we have to learn about the segmentation workroom, okay? So I will go through it. Let me show you what that is and what I mean by that. Let me open the SAND dataset. Again, just a very simple dataset. The focus here is on the frame, on the segmentation tool. Again, I have the SAND dataset here that we had from earlier. Same thing. Now, instead of creating modules here to segment this thing, I'm gonna click the data object and click here on the menu on top. There's a segmentation button. If you click that, it will actually bring you to a different world, right? So it's a different user interface. And it's called the segmentation workroom for a reason, because it's focused on segmentation, right? Let's say doing more advanced segmentation, all right? So let me just give you a walkthrough of the different windows. So on the top left here, you can specify your input image. Your output image here automatically gets created, or you can create a new one. A patch set, we'll use this later in the deep learning part. For now, it's empty. Here you have a list of materials. So you have exterior, that's assigned to the value of 0. Material 1, that's assigned to a value of 1. And you can add new materials, let's say, and you can rename them. Let's say I want this air and sand. So this will have value of 1, value of 2. You can change the color. But right now it's empty, right? You don't have anything. And here on the right, you have your 3D view. It's empty. And the 2D view of your data, right? So you can change the slice number. You can change the contrast as well if you click here. It's a bit dark on the screen, so let me adjust this a bit. All right. And then on the upper part here, you have tools that allow you to create what's called a selection, right? So it's basically highlighting parts of your image. So the most basic tool is a brush. So if you click on brush, and if you go on an image, if you hold the left click and move it around, it's a paintbrush, right? So it allows me to manually segment my data, right? So kind of worst case scenario, this is something you can do. So there's shortcuts to fill stuff and so on. If you want, this is the worst case scenario, right? Of course, you don't want to do this, but I'm showing you the tools because we will use these tools a little bit later. So you can do this manually, but of course, we want to avoid it. That's why we have tools like the thresholding and so on. But for now, this is important. So I highlighted stuff. It's shown in red. Right now, it's just a selection, right? You just highlighted it. But now I want to add it to one of my materials. So I click on sand, and click add to. Once you click that, it's now colored in pink, which is the color of sand here. Let's change that to blue. Right, so now you've added it to sand, right? I can do the same for air, yeah, and click air and then add, and so on. Of course, you don't want to do this manually, but I'm just showing you, if you are gonna do manual, this is how you do it, yeah? And we're gonna need this for the deep learning later. There's other tools, I will skip them. There's an eraser, obviously to erase the stuff. You can change the size of your brush and so on. You also have thresholding here, right? So if I click thresholding, you can set the threshold value here, yeah? And you can click create selection. It highlights the stuff. So it's similar to using the thresholding tool in the project window. And you can click the sand and say add to sand. Yeah, okay. There's other tools. There's watershed. There's texture classification. I'm not going to go through those today. We actually have those in previous workshops that are in our YouTube page, so you'll have access to that later. What I want to focus on is the deep learning tool. And that's over here. So if you click here, it says AI-assisted segmentation, right? So this is basically the deep learning tool. Now, before I show you how to use it, I just want to show one slide. Right? So for those who are not familiar with deep learning, I will not discuss the theory of that. The cool thing is that you don't have to be a deep learning expert to use deep learning in Avizo. Right? So the idea is you have your image data, and Avizo has some built-in deep learning models. And all you have to do is provide some initial training data, right? That means, I don't know how you generate this training data, most likely manual, right? The way I showed you, you use the brush to manually select the stuff you're interested in and feed that into the model. And the model will try to learn how to segment the stuff you're interested in, right? That's the big picture idea, right? The idea is you provide a few samples, and then the deep learning model will do most of the segmentation for you, okay? Of course, the result might not be perfect, but at least it will do a lot of the job for you, okay? Sounds good? So this is the big picture. As I said, you don't have to be a deep learning expert, but it's good to know, right? Like the underlying theory. All right, so first, before you do the hands-on didn't provide it to you because I'm not allowed to. But it's basically a membrane, okay? It's called a zeolite structure. I'm not an expert, but it's apparently called a zeolite structure. Okay, volume rendering doesn't look good. But if you look at the slice, it looks like this, all right? So it's a membrane material, and basically there's two things that are interesting to the user. Those are these like circular shaped things over here that are like repeatedly appearing, and the pores that are like very small around those balls or spheres, okay? So for now, I will just try to segment the spheres, right? And as you can imagine, if you just try to use thresholding, it's very difficult. It's very difficult to get the balls, okay? It's very difficult, okay? I hope that's clear. So in this case, I would use the AI segmentation tool, right? So let's go ahead and do that. I will go to the segmentation workroom. And for this one, I just want one material. Let's say I'll call them spheres. And let me pick a good slice. So I will zoom in here. How do I zoom in? Oh, okay. Okay. And yeah, I will pick like a nice region. Nice in terms of I want parts where the spheres are clear and maybe easier for me to segment. And I will now do it manually, right? So the nice thing about the AI tool is that if you're lucky, you only have to do this a few times, right? Which is amazing. I mean, the first time I tried this, I was mind blown by how well it worked, okay? Of course, this is not perfect. Yeah, and I'm not an expert. Maybe I'm doing some things wrong. But let's say this is good, yeah? I selected those. I add it to my spheres. Boom. And now, let me change the color. It's still not clear, but anyway. Now what I'm gonna do is I'm gonna create a new patch set, right? And here you can pick the size of your patches. Now, if you're familiar with deep learning, this is basically the size of the images that are fed into your network, right? For training and prediction. If you don't know what that means, that's fine. We can talk about it later, but for now, let's say I pick size 64. And all you have to do now is click this button. This is Add Patches. Let me just zoom out a little bit if I can. Yeah, all you have to do now is click on the image where you have your sample segmentation, and also outside where you don't have the label, okay? Because you have to show the model as well in how the non-sphere parts look like. So I'm gonna segment more here because this will be fed into the model as well. Yeah. And of course, it's up to you how much training data you want to generate, right? Like, do you want to spend minutes doing this, hours? It's up to you. So now I have my patch set. I will click this AI segmentation module. There's only, let's say, 2 numerical parameters you need to set. The number of epochs for training. So the higher this one, usually the better, but the longer it will take for training. And then the deformation strength, if you're familiar with data augmentation, this kind of tells Avizo how much you want to deform the data for training, like stretching, rotating, etc. And that's it, right? So for now, let's say I'm happy with that. Let's see what it gives me. I say compute label. And I say new, so create a new label for the result. And let's see how well it does. Okay. I mean, this can be totally bad or totally good, but let's see. And that was what? A few minutes worth of effort, right? So it's done. Of course, oh no, it's not. So you see the training here. The speed of training, of course, depends on the size of your data and the GPU you have. So there's requirements, of course, for you to run this. You need a good GPU, and I think it only runs on Windows. Yeah, so now it's done. I'm not sure if you can see it, but the green, I mean, let's change the color. I hate the default colors here. Okay, it's not super obvious here. Actually, it looks good. So let's go to the project view. So you can always go to project view and look at the result. Okay, let's look at it in 3D. Okay, this is the result, yeah? So you say zeolite.ai assisted segmentation. And if I look at this in 3D, it's amazing. I don't know if it's obvious to you. Wait, what's the best way? Let's do this. Let's do this in 2D, right? So you can do an ortho slice. This is another trick. If you load an ortho slice and then click this here, add color wash, you can connect the segmentation. Look at that. Look at that, right? So this is the original scan, and we overlay the result of the AI segmentation. I don't know about you, but to me, this looks amazing for a few minutes worth of work, right? Like, to me, this was mind-blowing the first time I did it. And the thing is, I appreciate it a lot for this project because I spent hours trying to apply filter and thresholding, right? Just to get something. But after 5 minutes of AI segmentation, I get this. Of course, it's not perfect, but it does, I would say, 80% of the job pretty well, okay? And it's nice to report that in your paper, right? Like, oh, we used deep learning to get something. But I hope you agree that this is just amazing, right? And yeah, you can use this for your project as well. Just a quick note. Of course, it's not magically just gonna work for your data. Sometimes it doesn't work, right? So we've had cases where nothing works, so we ended up manually segmenting everything, right? And later I will show you how we can help you with the manual segmentation, all right? Okay, so that was it for the example. And now, for those of you who have the segmentation workroom installed, you can open this cryo-EM example. This is actually from Thermo Fisher, so the Avizo developers. And you can find this example online, so I provide the link here. The slides will be uploaded to the workshop website, so you can go back to this. But if you go to the data folder, you should find this cryo-EM segmentation zip file. If you unzip that, you actually will find a PDF and a video, super nice video that shows you what I just did step by step. for this cryo-EM data. So the purpose of this is for you to now try to do this on your laptops. But instead of following me, you can just follow this video. There's also a PDF version. Yeah, I recommend the video. And there's an Avizo project here. If you just double click that, that already gives you all the results. Yeah, and the final result looks like this, right? So this is the fibers, these things, I don't know what they're called, already segmented, right? So this is the final result. If you're just interested in looking at the final result and the training data that was used to get the results, you can just open that project, okay? So, okay, I think for the sake of time, I mean, let's do that later on because we don't have much time. So you can stay, for those who are here in person, you can stay and do this, and I'll be here to guide you through it if you have questions. But for the sake of time, I will move on to the last few slides so that we can maybe end the stream on time. So yeah, so far, what did we see, right? So we've seen how to use Avizo to do basic visualizations, basic segmentation, and a little bit of advanced segmentation. But as I've said, there's different ways to reach your final analysis. There's also different modules, there's different tools. Personally, this is kind of my workflow, right? Personally, if someone comes to me with a new data set, they tell me what their data is about, they give me a copy, they want to segment something, this is how I do it, right? So first, I inspect the data, right? Just take a look in Avizo, right? Use the orthoslice, volume rendering. I check the histogram, maybe there's like interesting peaks, there's interesting things in the histogram. So just inspect the data, get a feel of how it looks like. Is it noisy? Is it easy? Is it difficult? For me personally, and for you eventually, you'll get experience and figure out that if you need to apply a filter, right? And usually I would try median filter, bilateral, non-local means, unsharp. If you don't know what these mean, it's fine. You can read the documentation or approach us. Just try things out. And the goal is to apply a filter so that when you apply the other tools, it's easier to segment, right? Like thresholding, which is the main thing we use today. There's other powerful tools like Watershed. I didn't talk about it today, but you can check it out in our YouTube videos. Also texture classification. This is kind of older machine learning based stuff. If these don't work, or from the first look, you already can tell that it's so difficult, I need AI, then use the AI tool, the one we just showed today. If that doesn't work, try other tools. So there's actually other tools other than Avizo that we support in the VizLab. There's another tool called Elastic. You can Google it. It also has AI segmentation tools, all right? I'll show one slide about it later. Elastic also has a texture-based classification. Try that as well. And then there's more complex deep learning modules in Avizo. I will show a slide about that. You can try that as well. Try other tools. Worst case scenario, you can do manual segmentation. That's worst case, yeah? Because that means you'll have to spend hours, days, weeks segmenting your data. And that's what we want to avoid. If you don't know what to do, you can always reach out to us, right? This is the, I would say, best thing to do. And then we can help you. So as I said, Avizo has a more complex deep learning tool. And here you really can play around with the parameters of the deep learning model. Number of layers, number of epochs, the learning rate, the optimization algorithm. So there's different techniques or different parameters you can tweak, but This is more advanced. Reach out to us if you want to use this. As I said, Elastic, another tool. It's also described in our YouTube videos. There's other tools like Fiji. I think this is used a lot in the imaging, the microscopy lab here. And finally, if you eventually realize you have to do manual segmentation, we have this pen and tablet interface that would make it so much easier to do manual segmentation instead of your mouse. Okay, so we have this this facility that you can book through our booking website, and I've had 2 users who actually use this to segment their data manually, and they spent 1 to 2 weeks manually segmenting their data, because in the end, for example, their data was like 200 images, and the material was 200 images. If you spend, like, let's say 1 hour each, right, it's not so bad. And then eventually you get a perfect segmentation, right, because this is manually done by you. Again, this is worst case scenario, but it can happen, right? So that's the end of the workshop, at least for the stream. You guys can stay here. A few things I want to say. First of all, thank you. This is our website, and this is our email. This session is free. Please give us some feedback. It's a one-click thing. Just 5-star or whatever. And if you have comments, right, if you think we can improve the workshop, please give us some comments. We really appreciate that. And I just want to thank my colleagues, Tom, for helping today, and James for setting up everything and monitoring the stream. And yeah, thank you very much. For those who are here, please, if you want to stay or have questions, if you want to start collaborations, please, we love that. Okay? Thank you.